It is a pleasure for me to present Nir Sade. I know Nir Sade for a long time. He's right now having a position at the University of Tel Aviv with his own group. But he has been doing a very eclectic, I will say, transition in between his PhD, his postdoc, not only in plant physiology, but also plant biology, biophysics, genetics, and a little bit of metabolomics also. Yeah, yes. In Europe, mostly, one part in Europe and also one important part in the United States, which is also a way just to have integration from different perspectives. I mean, the way Americans work and the way Europeans work, and I think he has the opportunity to feel the physiology in one way that nowadays is like a little bit loose, in terms that people is more concentrated in the genetics and the cell biology and the molecular biology. So he has an open mind, and I think if you read or follow his publications, he's also exploring new models in plants that are very important in the terms of crops and culture, and I think it's challenging, so I appreciate a lot for him joining us today.
Thank you, Gabi, for inviting me. Thank you all for coming. And of course, I was here first time 15 years ago, so it's very nice to come back to Argentina. I was here as a mochilero. So I know all Argentina. 5 months I spent here, but not as a biologist with the backpacking. So it's nice to come back to Argentina and see the other side. I only know tents in here and all this hiking. So thank you very much. We will talk today about one part of my lab research, which is concentrating on the hydraulics and gas exchange conductivity, and how gas exchange tracks root hydraulics conductivity, which in turn we think is dominated by plasma membrane aquaporins in the new model plant Setaria Viridis, which is a model plant both for C4 crops like maize, sorghum, and is a very important model plant also for cereals. So just a quick introduction where I'm from. I'm from Tel-Aviv University. It's a kind of big university. Like here, we are sitting in the middle of the city. So of course, we have lack of growing. Like everybody, you know this, so we have the same problem. And I'm located specifically in, of course, in the Department of Plant Science, but in a specific institute called the Institute of Cereal Crop Improvement. It's an institute that dedicates most of the research for cereals. Okay, and I'm sitting in this institute. It's a very unique institute because we have a huge amount of species of germplasm, cereal germplasm, all native to Israel. So Israel is very unique. It's a very small area, but very different in the ecology. So it's like microclimate. So you can see we have tens of thousands of species, specifically of wild wheat. I don't know if you know the wild wheat, the original wheat, is coming from Israel. So we have a lot of wheat growing in the wild, and it's a huge resource from genetics perspective. And we have a lot of oat, rye, brachypodium, barley, all sitting in our institute, and we are trying to utilize those germplasm by doing sequencing and other stuff. Okay, so this is just a quick introduction from where I'm from. And now if we start a quick introduction for the lecture, so this is just something that you probably most of you know of how water moves through the plants. So water are absorbed from the root system, where they go up through transpiration stream by hydrostatic pressure gradient, by differences in the water potential. They go inside the roots radially, then they go axially to the xylem, again radially into the leaf, where they evaporate to the atmosphere through the process called gas exchange from the stomata, which of course at the same time, when water goes out, carbon dioxide go inside for photosynthesis. So although this is known, the signals that regulate this specific procedure of the gas exchange is not really established. Some claim that it is an hydraulic signal coming from the roots to the shoot. Some claim that it is a chemical signal like IBBEA. Some claim that the stomata are responsive to both atmosphere. Some claim are coming from the roots, so it's a very complicated system that they don't really know what really regulates the process of gas exchange. And from the perspective of the gas exchange, we can divide the plants to two different, very roughly, yes, to two different groups, one called isoidic and one called anisoidic. The basic differences between them in terms of the gas exchange is that the anoisoidic usually have an intrinsic higher gas exchange capacity, so higher transpiration. They can maintain more open stomata, allow and continue for gas exchange, compared to isohydric, which are more, as we call, conservative. They really regulate the stomata tightly. And if you think about this, it's two different risk-taking strategies. The anisoidic, kind of more of a risk-taking. He will allow the stomata to remain open. He will gain gas exchange, CO2, but can also suffer strongly if there is a strong stress. Okay? While the isohydric, on the one hand, is very conservative. He will close the stomata very fast when stress comes, but he will also pay for productivity because there will be less carbon dioxide fluxes coming. Okay, so this is kind of like a model explaining the anisohydric and isohydric. You see the transpiration. And if we go to the root perspective of the water flow, again, we can look at different pathways of how water moves through the root system. Total the movement of water from the root system called root system hydraulic conductivity, which is the potential or the ability of the roots to facilitate water per unit of area. Okay, it's how good the water moves through the pipe system, basically. And altogether, root system hydraulic conductivity is composed from axial conductance, which is mainly the xylem. Okay, how the water moves through the xylem system, and this is kind of unregulated system, right? I mean, it's dead tube. It's basically dead cells, so you can not regulate it by proteins. You can regulate it by the amount of xylem and xylem development, of course. And there is another aspect of fluid hydraulic conductivity, which is the radial water movement. It's here. And this is also divided to different pathways. One is the apoplastic pathway, which is the blue here, where the water moves via cell walls. And one is the symplastic pathway, which is here, which by the water moves either by plasmodesmata connections or by transmembrane connection. And it's very hard to differentiate between those two. Even till today, we don't really know how to differentiate. Okay? Altogether, all those pathways define what we call root hydraulic conductivity, okay? Which is how good the water will move to the root system. So in our lab, we started to develop some system to measure grassroot hydraulic conductivity. It's a kind of a simple system. Basically, we expose root system to different gradient of pressure. Basically, we push the water to the root system and we collect the exudate in different pressures. Okay, and then we calculate the flux, how good the water moves, basically the flux of the water per unit of area or a unit of dry weight. Okay, and this is how we calculate root hydraulic conductivity. Again, I remind you that once we do this measurement, we get the whole root hydraulic conductivity. We don't know the different component. Okay. Okay, so when we started to approach this system of studying the relationship between root hydraulic conductivity and shoot gas exchange, we decided to work with a new, an emerging model plant called green millet, Setaria Viridis. We decided to work with this because it's, again, a very established model for C4 plants. So there was a lot of genetic resources, genome available, and a lot of molecular stuff developing, but there was no physiology at all. When you look at the literature, nobody really started to address things like hydraulics and physiological measurement in this model. So we decided to attack these specific questions. And again, it's a relatively convenient plant to work with. And I say relatively because it's not Arabidopsis, of course, but relatively okay. You have a short life cycle, small structure. It's a self-pollinated plant, so you can collect a lot of seeds. The genome is relatively small and relatively simple cultivation requirement. And those are many papers came out in the last year related to Setaria Viridis and some resources. Okay, so how we approach? We first wanted to find, in order to learn, to ask the questions, we wanted to find two accessions that have contrasting behavior in physiological measurements, so we can study in deep why they are differentiated. So we screened for several different accessions. we identified two different accessions from different areas of the world. One we called SAJ, one is ZAJ. We started by characterizing their transpiration, whole plant transpiration. So here we utilize a lysimetric gravimetric system where we scale the plants constantly, and by scaling the plants, basically we can calculate it how much water has been transpired. If you cover the evapotranspiration from the soil, basically the loss of weight is equal to the amount of transpiration per day. And we have a big system in Tel Aviv that we built, and using this system we quantify and we're able to identify two accessions that are different in their shoot transpiration rate. And it was very repetitive and very robust. So we took those two accessions and we asked, okay, we know there are differences in the physiology of the transpiration, whether root hydraulic conductivity might be a regulator of the shoot gas exchange of S. viridis, and we specifically asked about those two accessions as a model. So we started by measuring root hydraulic conductivity, and other measurement we build because it's easier to work and measure root hydraulic conductivity in hydroponic system. We first went to hydroponic system. We established a hydroponic growth system, and we quantify again using gas exchange, LICOR. We quantify in the leaf level the transpiration rate and the photosynthesis. And again, as I showed you before, the gas exchange of the ZAJ genotype was constantly higher compared to the other genotype. This was also accompanied by a lower leaf water potential at midday. So we quantify the leaf water potential, and because there is higher fluxes through transpiration, the leaf water potential of the higher transpiring accession was lower. Okay? Gave us another proof that there is stronger outflux of water coming from this plant. Most importantly, I think, is the quantification of the root hydraulic conductivity here. When we quantified the root hydraulic conductivity, we saw a very significant differences between the high transpiring genotype to the low transpiring genotype in terms of the root fluxes per root area, which give us some indication that there is a good correlation between the root hydraulic conductivity and the shoot gas exchange. So I told you before, we have root hydraulic conductivity, but now we have different components. So how we really digest those different components? So we decided to address those components by different measurements. The first component that we wanted to quantify is the axial conductivity, basically what is the differences in the xylem. And we did this by basically doing a longitude. We did some cutting of the root system and we quantified the xylem area. We didn't identify any major differences in the xylem and the amount of the total xylem between those two accessions. Now, this doesn't mean that xylem does not contribute to root hydraulic conductivity. It just means that in those two accessions, this is the not main component that contribute to the differences between them. Okay? So after we eliminate the axial conductivity, we went and tried to quantify what we call apoplastic barriers. So in order to do so, we decided to quantify the main barriers for a apoplastic flow, which is the suberization of the endodermis, as you can see here. By using specific dyes, we quantified the roots endodermis suberin and quantified, and we couldn't identify any major differences in the suberization between the SAJ and ZAJ root system. In here, we use propidium iodide to get the background of the whole cells of the root system, where you can see that indeed most of the barriers are in here, in the exodermis of course, but also in the endodermis here. So now we have two contrasting accessions with clear differences in the gas exchange and clear differences in the root system hydraulics. We cannot identify big differences in axial conductivity or in apoplastic barriers. So we went and attacked the other pathways, which is the transmembrane pathway. The transmembrane pathway is mostly defined by diffusion through membrane, and one of the main regulators of transmembrane pathway is aquaporins, which has been known for many years, and of course Gabi contributed a lot to this area. And as you can see, when aquaporins are present in the membrane, the diffusion of water is much faster than just simple diffusion. So luckily for us, when we started this research, somebody did a phylogenetic tree for the Setaria Viridis aquaporins. It's usually kind of a big family divided by different subfamilies, MIP, SIP, TIP, and PIP. I will not talk in length about the different family. I will just say that we concentrate on the PIP subfamily because it's the family of the plasma membrane where most of the regulation of water flow from the membrane is through the plasma membrane. So we decided to focus on this family. And then we wanted to ask, okay, how can we quantify or get some correlation measurement that imply for involvement of aquaporins? So we did two kinds of experiments. In the first experiment, we quantify the root hydraulic conductivity in the presence of aquaporins inhibitors, chemicals. I would just want to emphasize that this is sodium azide. Those are inhibitors of aquaporins, but are not super specific inhibitors. Okay? They're also doing a lot of other stuff in the plant, but this is what we have today. So as you can see here, under control condition, again, very consistent that the ZAJ have higher root hydraulic conductivity, but upon exposure to aquaporin inhibitor, both accession dropped drastically and became the same, which again indicate that many of the differences between two accessions are attributed to transmembrane pathway. The second experiment that we did is try to quantify the cellular water flow. So by doing so, we developed a system where we extracted root protoplast, isolated protoplast from root system, and exposed them to hypertonic solution where the water will flow into the cell will swallow, and we try and quantify the swallowing of the cell. And basically by doing so, we calculated the kinetics of water flow. If one cell has theoretically, higher activity of aquaporins, the water will flow faster than just a simple cell without any. So if you can see here, I hope that the movie will work. This is just an example of a root protoplast. You will see how the protoplast is swelling. I think you will see it. 1 second. You see? So there is a swelling of the protoplast. You extract the protoplast and you measure, and by doing so you can quantify the cell water permeability. When we did this for the two accession, again we identify a clear, significant higher root cell water permeability in the genotype with higher root hydraulic conductivity, giving us another indication for the involvement of transmembrane pathway. Okay, so this is perfect, but there are many aquaporins. So how we continue? Up until now, it's all correlative data. We wanted to really go deep and try to get some direct data. In order to do so, you have to use mutants and other methods, and for this, you have to have candidates. So we decided to basically look for our candidates by doing expression analysis. Now, of course, I'm aware that expression analysis is limited. There is protein level, but this is the best method that we have to try and differentiate between different isoforms. So we quantify the different aquaporins in the roots between the two accessions, and we identify several isoforms that were higher in the high transpiring accession. You can see here a very nice differences in PIP25 and differences in PIP15 and PIP16, which gave us some indication to do an educated guess of which are the isoforms that we want to explore. As a test case, we decided to continue with PIP16, first of all because I personally think that PIP1 are understudied and are more interesting to go with because they are not really characterized water channels, basically. And second of all, this specific one was almost non-studied at all, and also it's unique for Setaria and maize. There is no PIP16 in Arabidopsis even. So we wanted to continue with this as a test case. Okay, so then we said, okay, let's start to learn what is the role of this PIP16. And looking in the literature, of course, everybody knows this lady in here, has been suggested that aquaporins PIP1 can be regulators of PIP2. So basically, PIP1, the few measurements that was done were showing that are not really a good active water channels. So how do they change water permeability? So what has been suggested that they might bind to PIP2 because aquaporin is a tetramer for monomers. They might bind to PIP2 and basically regulate them. And by doing this binding, heterodimerization, they basically help the PIP2 to become better water channels. So following this hypothesis, and some people that even show this, we wanted to see if this is the case in Setaria Viridis. So we isolated the PIP16, fused it to GFP, and we isolated several PIP2 and fused them to CFP, different color. And we wanted to see what happens if we co-express them together, whether they one affect the other localization. So this is the result. You can see here, very interesting result. When you express the PIP16 alone, now I would like to emphasize this is a hydrology system. This is Arabidopsis protoplast, okay? Because it was very hard to, it's transient expression. It's very hard to extract protoplast and also do transient expression on the protoplast. The efficiency is very low. So we extracted protoplast from Arabidopsis. When you express the PIP16 alone, you clearly see a localization that is resembles ER, okay? We also use specific marker. You see here the blue is the ER marker, and you can see a clear co-localization, meaning that the PIP16 predominantly go to the ER when expressed alone. However, when you express the Setaria PIP16 together with either PIP21 or PIP25, you clearly see a change in the distribution of the aquaporins. So